So really the easiest example that I have for this when we talk about it is really the hemolytic disease of the fetus and newborn. So in this instance, this is where we have mom. So there's mom. And we'll make mom look like this. And inside mom is this absolutely adorable, cute little baby in there. Okay? And what unfortunately can happen is mom can make an antibody. Mom will have made lots of antibodies, okay? Because don't forget about all those antibodies that we have out there to our chicken pox, to EBV, to the flu, etc. Okay, so there's lots of antibodies in there. Okay, and a lot of those antibodies can cross the placenta, and they're designed to protect the baby, so that's a good thing. But what unfortunately can happen is if the baby on their red blood cells down here, I'll draw some little red blood cells in the baby. Their red blood cells have a different antigen than the mom, okay? And then mom, unfortunately, can form an antibody to those. And if mom has an antibody, okay, to that antigen specific on the baby, that antibody can cross the placenta and attach to these red blood cells. Okay. This is what causes hemolytic disease of the fetus and newborn. So what we would do with the DAT at that instance, after the baby's born, we would collect blood, and it's typically from a cord blood. Okay. That's really easy to obtain after birth. So they send the cord blood down to be tested in the laboratory. I don't know about everyone else, but we frequently get them in pink top tubes, EDTA. Okay? That was just to make them look different than hematology tubes. So we would get a cord blood in there. And so it would be full in there, and a lot of times they usually were not real pretty specimens. Okay? And so we would test it for a DAT to see if mom had any antibodies that had sensitized. And so what we would see, we'll go ahead and now erase this real quick, and we'll draw this in here. So if mom had formed antibodies to the baby and they had crossed and sensitized those cells, my circle doesn't want to work real well at the moment. There we go. Okay, so we would find those. And so these are IgG antibodies. Okay, and remember, IgG antibodies are smaller than M, and so we can't detect them at immediate spin. Okay, so for DAT, we get a sample, and we can do this on adults too. So what are some instances? So we said HDFN was one, HDFN, hemolytic disease of the fetus and newborn. Okay, we can have hemolytic anemia occurring for multiple reasons inside of a patient. Okay, so we'll just write hemolytic anemia. We can have an autoimmune hemolytic anemia occurring. And these can all cause positive DATs. So with just a hemolytic anemia, what's occurring here is you have some sort of antibody attaching to the cells. And it could be, we talked about the KID antibody. That's a delayed hemolytic anemia. Okay. Because that antibody likes to go below detectable limits. Patients transfused because we don't know they have an antibody, and then that antibody attaches to the red blood cells and undergoes, in that case, it's going to be extravascular hemolysis removed by the spleen, the reticuloendothelial system. Okay? In an autoimmune type, this is where the patient makes some sort of antibody against their self. And that antibody then attaches again to those red blood cells inside the patient. Okay. The antibodies attach. They have varying specificities. Okay. They attach and again cause hemolysis a lot of times through the reticuloendothelial system again. A lot of times it's extravascular. There are some rare instances that it can be intravascular, and that can be very significant. And one thing to remember in an autoimmune type incident, we really, the goal is to fix the underlying cause. Many times, if you transfuse this patient, okay, if you transfuse them, that'll actually exacerbate the situation and cause more hemolysis. In this instance, you're really going to want to communicate and work with your physician and determine the significant need for the patient and if they can wait a little bit and try to fix that. But those are some reasons why we would have positive DAT. Okay, so let's get back to our specimen up here and take a look how we are going to test it. Okay, so now we have our specimen down here. We have it in the laboratory, and one thing we want to really remember is we want a fresh specimen. Okay, we want, whoops, I picked the wrong one. We want a fresh specimen, okay? We don't want to pick one out of the refrigerator, okay? That's bad. A lot of times our complement tends to fall off. The cells, I say fall off, but it's this constant equilibrium, so to speak, in there, but it will detach and we get a false negative. Another one is we want to do EDTA, okay, specimen for this. The red top clotted tubes, they're prone to some false positives, okay, so we don't want to use those anymore either. So we have our EDA specimen. We want to, again, make a fresh cell suspension. Okay. We don't want to just use the cell suspension that's been sitting there in our rack that we used for the ABO and RH. Again, if there's something on those cells as it's been sitting there in saline, it has a tendency to detach, and we don't want that, whether it's an antibody or the complement. Okay. So once we have this specimen, we're going to take this specimen, okay, and we're going to make our 3 to 5% cell suspension. Okay. Here's our cell suspension. So now, once again, we're going to have these red blood cells in here. And in this example, I'm just going ahead and I'm putting the antibodies on here so that you can see them. Okay. Whether you go through, your immediate spin, record that. Some sites, I remember incubating it for 5 minutes before. But the key to this is, we know this is IgG. Okay, this is a IgG at this point, okay, or complement, okay, which is what we're looking for. And we're not detecting that at immediate spin. Okay, so what do we need to do? So we're going to put a drop in our tubes. Okay, so here's our tube. We're going to put 1 drop of cells in here. We usually run a control with it because we want to make sure our cells are just not agglutinating for any old reason. And we want to wash them. Okay, we want to wash it ×3. Okay, sometimes more if it's a cord blood. Follow your policies at your facilities where you're going to be working. Cord bloods, we want to wash away that Wharton's jelly. Okay. Wharton's jelly will cause a false positive in a cord blood. Okay. So we want to make sure we wash it. And once we wash it, we're also eliminating any extraneous proteins or those extra antibodies that might be in here floating in the patient's plasma. Because we're going to get a little bit in there when we make that cell suspension. So the washing will eliminate that, and we will just have then the patient's cells in here. Okay. And in here, this is our control. Okay. I'll make my circle a little bigger. And there's antibodies on there. Okay. And there you go. So we've washed it, and now at this point we're going to add our, usually we start off with poly Coombs. Okay. We're going to add our drop of that into our DAT. Okay. This is going to be our control. We're not going to add it into our control, or it wouldn't be a control. It's the same type tube. And usually you're going to add maybe a drop of saline in here to keep the suspension going. Okay. And again, this is just because we want to make sure these cells are not just spontaneously agglutinating. So if there's antibodies attached to those cells, let me go back to the right color. Our AHG is going to cause that lattice formation, remember? We described that before. And we'll see agglutination, because there's antibodies coated on there, and that AHG is bridging that so we can see that agglutination. And that would be a positive DAT. Okay? Now we want at this time, our control should be negative, because that's what our controls in blood bank. These are negative controls. Right? Now, what do? We have a positive DAT. One thing we need to think about, do we care? Do you need to identify it between IgG or complement? And your facility may require you to do that. So you would set up 2 tubes. Okay? Just follow the policy where you're working. 2 tubes with those cells again in them. You're going to wash them, and instead of just adding the poly, you're going to add 1 drop of monospecific. Okay, so up here we used a poly, and that's what this right here shows you. This is poly. Down here we're going to do mono, which means 1 reagent has IgG and 1 reagent has complement. So we'll put 1 drop complement in this tube, anti-IgG in this tube. So remember, it's anti-IgG and anti-C3d, or complement. I tend to abbreviate it like that. Okay. So now we're going to determine which one of these agglutinate. And so if this one's positive, then it's IgG. On the other hand, If this one's positive, it's complement. And which antibody is the best at binding complement? That's an IgM, so we can conclude it's probably an IgM antibody that's causing the problem at that point in time. Okay? And so that's kind of a quick little review about your DAT. All right, thanks a lot, guys. Hope that helped. Signing off.